Started. Awesome. Welcome everyone to the Biopython event, sequence alignment. This will be our last Biopython event series. We have been learning about what Biopython is and how we can pull data from databases, curate it, put it together in a file. And today I'm going to wrap it up with the alignment, which is the last part. You can get a pretty big picture so far that if you can get a file from NCBI and then put it together in a big file and then do alignment, that's practically the entire project that you'll be asked in some of the classes. I hope you guys like this last event, and I shared the collab notebook on GroupMe. So we'll get started as soon as everybody is ready to go. Let me know when everybody has it handy. I am passing out. Yeah, so quick review on what we've been doing with Biopython. So last event at it. So if you missed last event, here's a quick overview. We looked at the unstressed module where we pulled information from NCBI databases. We looked at all the summaries. We then did a specific query on human data, a specific BRCA1 breast cancer gene, and we looked at different files related to BRCA1 gene. And then we put all of those sequences in one file using the write module, the seq.io.write. And then we parsed through it and we read how to get the sequences and all of that. And that was all covered in the last event. And now, once we get this sequences, now we can do the alignment to learn about the scoring and identity, similarity scores. That tells us the evolutionary of the sequences, so how two organisms are closely related. So that's today's also, okay. Perfect. So yeah, so overview for today's event, we're going to look at specifically align IO module, which parses through the alignment, which we get from, we can use web-based tools like ClustalW, Muscles, and all the other alignment tools available, interface friendly. You can upload your sequences and they'll return the alignment score. And you can download that file, and then you can load that file here, and then you can parse, read, and write. So if you open it, and then if you want to like splice it, you can do all of that in the alignment file, and then you can write a new alignment file and do a further analysis. There is we'll also look at how to manually write your own sequences. So if you have very few, let's per se, only 3 to 4 sequences manageable, then how to manually just create a file here using the seq record and multiple sequence alignment module. We'll look at that. And then there are interesting Biopython command line wrappers, which uses ClustalW, Muscles, Needleman, Watersmith, those algorithms to do the alignment. We'll look into that. They're all both global and local alignments. And lastly, we'll look at the pairwise 2 module, which does PAM, Blossom 2, all those different alignments. So we'll specifically look at Blossom 62, the substitution matrix, to wrap it up. Okay, with further ado, let's get started. So first things first, we're going to install Biopython to the collab notebook. Looks like it's ready to go. And then we're going to import this seq align. Here, in the last event, I didn't mention the help function. Help function is really helpful because you can look at all these different things you can do using that specific module. They give you detailed examples, so you can follow along there as well. I'm not going to get into that. There's a lot of things in here, but we'll touch upon a few of these. So first things first, we're going to get a alignment file, right? Because align IO is good for parsing and reading the data. So first we need an alignment file. So how are we going to do that? We're going to copy this link and go to this page, and we're going to look for an alignment. So let's pick a sequence. We're going to show up. Okay, I'm going to minimize this. And let's just look up a sequence. Let's say, since we've been using with BRCA1, right? So I'm gonna search BRCA1. Let's see if we can find sequences. There we go. So we have all these sequence alignment files now. Let's just work with the first one. It's BRCA1C terminus, BRCA T BRCT domain. So there, if you go here, we can download the file. So if you go to pfam, okay, sequences, there we go. So if you go to the sequences, right, there are 20,996 different BRCA1 sequences. If you go here, and then you can select the file format. So here, you can select the different file format you want. And then you want tree, alphabetical, all of that, and then gaps. So in alignments, there are different ways they're annotating the gaps. There are dots, dashes, or both. I'll just go with the mixed, and then generate, right? So it's generating a FASTA file. It's probably in TXT file, yeah. So for me, it was TXT file, but you can change this into, I'm going to change this again because I used TXT before. I'm going to use a FASTA file. FFA file, and then save it into your wherever your location is. And then what we're going to do is we're going to go back to our Google Colab notebook, and we're going to import that file. So now, how are we going to do that? We're going to go to this file folder, and we're going to upload this file that we just downloaded. So for me, it was here, FASTA file. Well, actually, it's still in TXT file, so I'm going to go just. So yeah, upload your file on here. And then once that's uploaded, you'll read that file using the align io.read function. And since we already have the file uploaded there, we give that it's FASTA file because we give the FASTA file here. There's Stockholm and MSF format as well. You can do that as well, but remember, if you do change different format, then make sure you mention that here. And then once you do that, let's run this. So I'm going to print the alignment and see. So yeah, so this is the alignment that we got from this PFAM NCB. It wasn't NCBI. What was this? Yeah, EMBL database. So we see that there are 80 rows. So there are 80 different sequences, and there are 149 columns, which means 149 sequence length in the file. And we see that there are all the alignments, and then we see the IDs at the end, and all organisms. So yeast, human, we can see canal, rat, human, right? And the specific different proteins or domains that goes along with that. Now, let's see if you want to only look at the length. Like only you want to simplify it and only look at specific things, right? So for that, we're going to let's look at the length of the sequence, which is 80, I think, because it already gives us the rows. And then you want to look at sequences, only sequences and their IDs. We can do this, so we can clearly look at specific sequences. Now, because when we're doing some research, right, we're interested in specific mutations, specific locations. So we're going to get into splicings, because suppose that we're interested in only looking at 15th position in this sequence and want to see if there is what kind of mutation there is. Before I go further, I want to quickly overview the amino acids, right? Because in humans, we are restricted to this 20 amino acids. There are many more, but we practically are revolving around this because this is mostly involving with human all the processes that happens in our body, right? And that's the abbreviations that we see are all here. So glycines for G, alanine for A, and so on and so forth. So that's what we're seeing here. And that's whenever we are looking at specific pathways in human genome, they always look at specific mutations where one amino acid is swapped or had mutated with another amino acid, right? And that drastically changes its effect on the physiological, whatever the function of that protein is doing. So that is the reason why, like, we have to look at the alignment and mutations that partakes. So let's look at let's say we want to look at specific um, um, place, right? So for that, we're going to get into splicing. So for instance, let's just do this. I want to look at sequences 3 to 7. So this is only picking the first 3 sequences. So if you're interested in first 3 sequences and want to analyze that further, we can do that. That way, I'm sure most of us are familiar with the splicing regular formats. There's few things that you can do. So here, for instance, if there are gaps, a lot of gaps. So this is giving the entire column, so column 15 across all the different. So if I print, where is this? If I print this, right? Yeah, so if you see a lot of gaps all across, these dots are represented by gaps. So if you don't want these gaps, we can remove them. We can splice them out. So for that, we're first assessing, this column 15. We saw that there's literally a gap across all the sequences. So we can splice that specific sequence out using this function, right? So we can use all the sequences up to 15, and then so that includes all the sequences up to 15, and then we can do sequences after that. So I later looked into sequences which showed that up until like 35 there were a lot of gaps. So I removed all the sequences and kept the sequences from 35 to 45. So now here I'm concatenating all the sequences up to 15 and then 35 to 45, and then putting it together into one file. So there, I'm going to quickly run that so you can see how that shows, right? So we can see now, right? There are a lot less gaps, and we have more closer look at the alignments, and then we can sort them based on their IDs. So it's always better. It's always a good habit to sort them so that we can look at the top alphabetically, and then pick the sequences we're interested in. So let's see on this. Right, so they're all now alphabetically ordered. So now we can clearly see there are BRCA1 genes, BRC1 genes. So that way, if we're doing some research questions, we should know that, okay, we're looking into this specific domain, then we can alphabetically just scroll down and look at this specific domain. Yeah, so now if you look at these files in the entire, if you open the entire file, you'll notice that there are these databases are referenced. They have referenced to other databases for secondary structures. So to look at that, we're doing this quick trick of looking at the records and then record.dbxref, which is database cross-reference. Guessing this is this just goes back to this. Yeah. Okay, so let's do. I don't know why this is. This should alignments in alignments. So let me debug real quick. Yeah, so it's just this file here, right? Practically, it's not reading 3 cells down. Interesting. Okay. Oh yes, 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 yes. maybe it's just not. There is no cross references in this file. That's probably why. Interesting. But yeah, if there were, then there will be the PDB references, secondary structure references. So if you're interested in that specific alignment, you can go into the other databases and you can look into more files through that. There's also, let's see if this works. Yeah, there you go. So you can look at annotations as well. So if there are more features, in this case, there's no more features, but if you're looking into complex alignments, then there will be more features and you can look at annotations that way. So that's a quick feature. Just simply looking at the record in alignments, just printing the records will give you all the annotations. Now we want to look at how to manually make the sequences, right? So we'll look at this seek record module, and we'll along with the multiple seek alignment module. In this, we're manually making the sequences, right? And then giving them the ID, because in alignment, we give them a sequence and then they have their unique IDs. So here we're giving three different three different alignments, and then we're passing the sequences using the seek record module and giving the IDs. So seek, so you have your sequences, you put it together, and they'll align, and then you can put it all together into one alignment file. Right. So if we print this, we should not. We can. Yeah. So it should only give you the class, right, and just what you just made, right? So all the sequences that you just made, and then once we get this, when we compile this alignment, our own alignment, we're going to write it into a file. So in this case, I'm using align io.write, and I'm writing it in a phylip file format. There could be different file formats. I think I definitely added a link somewhere up here where you can go and look at different file formats right here, file formats. There are several different file formats available. You can look at all of that and work whichever one best fits your project. You can change it. There are Philips, FASTA files, Clustal, Emboss. They all have their different formats. You can look at that. So let's see. Going back to the collab notebook. So yeah, we just created three multiple sequence alignment, and then we combine it together into a my alignment variable. Now we're writing. Now we want to write that into a file. So we would say I'm just going to name it my example.phy, which is a filler format. To run that, it should create a file here on the left in the folder. You see, and then now we're going to read that file. We're going to parse that file. So now note that the read format align io.read can only read one file if there's only one alignment. But in this case, we have given three different alignments in this file. So we have to use parse, and in parse, align io.parse, we're going to do a for loop. That's the only way we can read multiple sequences in one file, sequence alignment in one file. So we're going to do for record in alignment, then print the record seek and ID. So if we run that, it should give us all the IDs that we just wrote in a file, right? Next, we're going to do substitution matrix, which is the number of times two letters align to each other divided by sum of all the pairs. And this gives the true probability of mutations occurring throughout the period of evolution. So which we're interested in this because we want to see how specific positions been mutated, and you can study that mutation and see how maybe in 10 to 20 years, how that sequence is going to keep mutating, and we can see some changes in future that could affect physiological and metabolic pathways down the line. There are also two types, PAM and BLOSSOM, and you can look into these. I have just given a quick overview of substitution matrix and how that's made. PAM and BLOSSOM uses their own specific algorithms. They have their own different ways to make their own substitution matrix. Okay, so we are again making our sequence alignment, right? So it's like we did before. We're giving 4 sequences this time, and let's see, we print this alignment. So that's which what we just made is going to give that alignment. So there are 4 sequences. Now we're going to use simply the alignment substitution function, and that's going to give us a matrix. So if you see here, A to A to C, A to T, and this is a classic matrix. We see it's reflected on the diagonal. You can see the same numbers aligned. And then you can, if you're interested in specific PAM and BLOSSOM, Biopython gets into that packages, so you can look into that documentation and learn about specific substitution matrix that you're interested in. I believe in intro to bioinformatics, we learned in class some complex substitution matrices, and they were asked in question in exams too. So if you're interested in that, and if you want to quickly confirm your answers that you give in when you're writing your exam, because these are all take-home exams, you get open book, so you can quickly confirm your answers by quickly running these, because like the professor teaches manually, so we have to handwrite and make these matrices, but you can quickly put in your four nucleotide sequence in here and quickly make your matrix and cross check. That could be a quick way. Next, we're going to get into the bio-aligned applications. This was really fascinating to me, especially too, because here it's giving all these tools, and we can actually use these tools, cluster W, muscles, and Needleman, all these different tools on here on Biopython, and some of them at least shows that how to use them. If it's not compatible here in at least Collab notebook, but it shows that how to use them in a regular Python idle. So I'll get into that. So the alignment, as we know, or just an overview, there are 2 types, right? First time when it was first found alignment, we were doing pairwise alignment. It was simply 2 sequences, and then we were doing the alignment and seeing how many bases are same between 2 alignments and how many are different, how many gaps are there in the sequences, etc., like that. In multiple sequence alignment, one sequence is mapped to multiple sequences and vice versa. So it's dynamic programming practically, where multiple sequences are aligned across each other, like we're seeing up here, right? We have 4 sequences, and they're all like matching with each other to generate this substitution matrix. So all the sequences are mapped across each other, unlike pairwise, which is what is happening nowadays. So to simply import the application, bio-align application, we're going to just import that, and then from there are a lot of wrappers, the command line wrappers, which has all these different code. So this is Python based code, which is calling the tools itself, and then parsing and doing the alignment behind the screen. We don't really see how they're doing the alignment, but they're pulling all the tool information, all the command lines in behind the screen, and doing all the alignment. But we simply have to give our files and parameters that we want to do, what gaps we want to give, what score, and penalty, all those kind of stats. So we just have to give simple numbers, and then it takes all that information and puts it in the code in the background, and pulls. It's just alignment, it's standard error, etc., and then it gives out the file, an output file, and we'll look how that is generated here. So quickly, how many different parsers are available? So we are looking here, Clustal Omega, Clustal W. Clustal Omega is the latest version of Clustal W. DI Align, MSA probe, MAFT, that's also a popular one. Muscle is also a popular one. Prank, I've never heard of that, but that sounds funny and interesting. A prop code, prop cons. That's also an interesting one. But yeah, you can see all these alignment tools are available in Biopython that we can use. So I'm going to go through cluster W today, and there are others, muscles, and I've listed below, and you can work on that. So let's look at cluster W today. So we're simply going to import the cluster W command line so we can see it's right here. We're just going to import that from here. So import cluster W command line, and then we're going to create so output file. So if you just print this, right, it's going to give us the simple command line what's needed. So if you install this in Linux environment per se, and simply to run this tool, you just have to write cluster W2, and then you do this flag dash infile equals, and then you just give your input file, and it's going to give your alignment output file. So we're going to look at one alignment here real quick. Also note that we're using W2. That's the latest version. That was the regular cluster W before. There's also this help command I want to quickly show, because this is very resourceful, honestly. The help function gives you all the parameters that you need to customize your alignment. So let's see if I go all the way up to so it tells us, the background a little bit, how it's initializing the class and all the background code. And then it gets into specific data descriptions. If you're checking into clusterings and DNA matrix, if you're interested in that, you can create that. Gap separations, all these specific commands that we can do iterations, right? So this, you can read through this, and you can customize your command line. So that, how would you customize it, is simply just putting comma here and adding, whatever you're interested in. So gap equals, if you're giving gap some value, you can do that, right? So you can just add that simply by doing comma and adding more of that. So this is muscle. I want to get back into command line. Yeah. Okay. So now the thing about this is that in Collab Notebook, we can't load the cluster WEXE. So we first have to download this. So let's go to this cluster W page so we can download. We have to download the cluster W into our local server first. So for the Windows, we have to do the cluster W-2.1 win.msi. And then pick your spot. I already have it downloaded, and then you run it. It's going to take like 5 seconds max. So once you download it, you'll have to copy this code, because unfortunately we can't run the exe file on Collab Notebook. So you'll have to copy this entire code into your local Python idle, whichever you're using. I'm using Spider in my case. If you just copy and paste this into your local file, and then we'll also create a file. We'll also create a FASTA file. So I'll get into that, how to create the file. Example file. It's pretty straightforward. If you go to this link here, they already have data compiled for us. This is just an example. So if you look, this thing again, if you're interested in specific, your project, in this case, it's doing, what is this? Opuntia chloroplast gene. So if you're interested in your specific project, in our case, BRCA1 we were looking at, you could do seek IO, parse. You can do query, and then you could do BRCA1, and you can pull your however many sequences, and you can compile it into one file, and that would look something like this. And then you simply use this file and do the alignment. So in our case, we are using this, so we'll have to copy and paste this into a TXT file. So copy that. You'll create TXT. Right, Notepad. So paste that, and then you'll just save this file as, or whatever you want to name this. So this is Funshia, is it? Shia.txt. I can just give this file. Oh, actually txt. Okay, that's good. Okay, and then I'll just go here and I'll just save it. Right. So now going back to, so we have saved our data. So we have how many? One, two, back to this 1, 2, 3, 4, 5, 6 different sequences. And we're going to do alignment across the 6 different sequences. So I have it downloaded in my local file. Again, going back to my folder where I saved it. I saved it, where's this file? Right here. So this is the file that I just downloaded, right? It could be in FASTA format too, whichever one you want. Again, whichever file format you do. So just to note, quick note, that the file format we see here is in FASTA format, right? So sorry, you see here, it's in FASTA format. So we're going to make sure that it's in FASTA format. Again, did you guys get a chance to run your the clusterw.exe? If you run that, you have to give a specific path to that, and then R is for the string command. This is they wanted this to differentiate from the new line they used, the /t and /n, but we don't get into that. This is just know that for the string, to recognize it as the path to this program, you have to have R in the front, and you have the paths to that, and then you read your file. So clusterw command line, the clusterw.exe, that's the tool itself, and then in file. So in file equals the file name, right? So if I just simply run that, it's going to create. So besides that, we're going to assert the os file.is. So if there are any errors, if there's any missing values or errors, standard output errors, it's going to append that into the file. So those are just the cross-check. If there are any errors comes up and you have to debug, then it will be in the file, and you can look at that and you can go back and redo those things. So we're seeing here that there are 7 rows. So actually there were 7 files. Interesting. And then there are 109 columns, 109 sequences long. So we see that there is an alignment across each, and then if we do like some sort of tree, right? So align.io.read is simply reading the file, and so again, notice this is alignment file. It gives the output in the cluster format, and it's ideal to whatever alignment you do through muscles or Needleman that I'll get into next. It's a good habit to have it in cluster format because it's universal and it's readable. It's easy to look at, I guess. And then you can have it also created this dendrogram, you can say, which is a evolutionary tree kind of format. You can phylogenetic tree. You can look at closely related sequences. So if this sequences has organisms names, you can play around. There is more functions you can look into in Biopython package to make it look more fancy. If you want to look at humans, and then you want to look at closely related organisms to humans and to that specific genes or specific protein that you're interested in. So you can do that. So that's pretty fancy and handy way to quickly make your tree. So that's just one example that I wanted to share with you guys. Let's get back to our bio collab notebook. So if we go back to our multiple sequence alignments, next we have is muscle. Muscle is just the same thing, same as clusterw. The only thing is that muscle W also, so we can't run it in this collab notebook, so you have to do it handy again in the in the local Python idle. So I'm gonna have you, like, if you're interested in the muscle tool, then you can do the same thing that we did with our cluster W and run it, and that'll give you the output file, and then you can again read and understand what it's doing. Right. I want to get into a little bit about the embossed needle and water tool, which is the Smith-Waterman algorithm, which is local alignment, the Needleman-Wunschuk global alignment algorithm. So for that, it uses the gaps and opens. So if there are gap opens, there will be a penalty of 10. If this gap is extended, then there's a penalty of 0.5. And the sequences are like the scores are computed based on these parameters. So that is something handy, I guess you can use. I'm simply making here sequences. I just made two sequences here, alpha and beta, and I'm just simply writing it good old Python fashion way. And I'm giving the Needleman command line and simply um, generating a command line, right? I think it already had the output. Yeah. So again, if you have it in Linux format, right? Linux is very straightforward. If you install this tools in Linux, the Biopython gets into how to configure and install it in Linux um, environment. And as soon as you install it, all you have to do is just copy and paste this command line. And you, of course, you have to make sure that you have the file there. Both the files are there in the same directory, working directory, and it'll give you the alignment file output. So it's that straightforward. But I want to get into the specific example of pairwise sequence alignment, and we'll look at how it gives us the score and the start and end positions of the alignment. So simply, we'll just import the pairwise 2 module, and then we'll also use seek IO because the pairwise 2 is in the string format, and to read that we will need the seek IO module. So the sequences that we made earlier, these top two, alpha and beta, we're going to read those sequences, and we're going to align them using, so here we're using global alignment, so global XX. The XX stands for the first X stands for the match count is 1. So if two nucleotide in two different sequences matches, then it gives the score of 1. And if there is a mismatch, if suppose there is adenine in the first sequence and there's also adenine in the second sequence, so that's 2AA, so that's a match, so it's going to give 1. But if suppose the first one was A and second one was T, that would be a mismatch, so there would be no penalty, so it would just be nothing, so it would be 0. And same thing with the gaps, so there's no gap cost. So suppose there was adenine, thymine, and then blank, and then GC, guanine and cytosine, so there's a gap in the middle. So sometimes you can give the gap penalty, but in global alignment, there is no gap penalty. So in pairwise alignment, right? In local alignment, it could be a different case. So that, if you run this, it'll give me the alignment of 80 sequences, so that there is 80 different, that's the length of the sequence. And then if you look at only the first sequence, it gives us the entire sequence followed by the score of the sequence and the start and end position of the sequence, right? So it's 72 right now. So that it's very difficult to see. So the nicer format I found on here, that was this. It looks neat, and it also compares every single sequences with each other, and we can look at closely, you know, M versus M, valine versus valine. This is protein sequences, protein sequence alignment. So we're seeing all the abbreviations of amino acids here. Yeah, so now I want to keep using this same, but I want to look at the matrices now. So let's look at, now we want to, because the score, okay, remember that we did a just simple local and global alignment without giving any specific parameters of gap penalties and, you know, mismatches, all of that. So now to improve our score, to improve our alignment, we start giving the penalties and gaps. So we can start playing with that feature, and that I'm doing that with the Blossom 62 matrix here. With that, I'm quickly making a substitution matrix of Blossom 62. So here I'm loading Blossom 62. Again, there are many available. Blossom 62 is just one of the example. There's several different versions within Blossom, right? And so is in PAM. So in this case, we're making, once we make this matrix, so I already have it printed, that's good. Okay, so let's see. So the alignment, this is simply printing all the stats that is printed from Blossom 62, but here's the matrices. This is also one of the things that you'll learn if you haven't taken already Intro to Bioinformatics class. This will be one of the matrices that you'll be making in the class and will be asked probably in exam as well. So it uses this matrix to make to do the alignment of the matrix. So let's see the specific alignment across. So how is the alignment now after we do the Blossom 62 matrix? So I did not. Right. I want that. Why is this? Okay, let's see. About dimensions. Okay, if I just go for anything, that should give me nothing. Perfect. Okay. So if I just want to do simply the length, let's just do length. So 2, yeah. So there's 2 sequences, right? Alpha and beta. So that Blossom 62 was giving us the matrix, and now we're going to do the alignment using the Blossom matrix. So if you notice, the score drastically went up. If you notice from earlier, the score was only 72 when we used the global alignment with no gaps and penalty. But as soon as we added penalties, gap penalties, and extensions, gap extension penalties, we immediately saw that there was there was a drastic increase in score. And that's essentially all these different tools are doing. They're making different algorithms to improve the score so that you have a better alignment. And then you can use this alignment to do further analysis, right? So that kind of, I just wanted to expose you guys to all these available tools in Biopython, and get familiarized with this algorithms that uses and how you can improve your score and help you aid you into in finishing your projects for your classes. So yeah, with that being said, this is the end of this event. I have there's more evolutionary trees and all the other things that you can do. There's blasts in Biopython you can look into. It's a good package, and it's good that Pradeep, you suggested that we get into this, and we have some of the classes we learned in introductory Python classes. We did learn a little bit about the Biopython, but never learned about alignments and how to get a lot of sequences into one file and do analysis. So that's fun to look into, and I hope that throughout this Biopython series, everybody learned something new through each event and can apply this into the project and do something resourceful from this package. Any questions? I know I've been talking a lot throughout this entire event. Yeah. Oh yes, you mean the alignment file, or do you mean the? Okay, yeah, the alignment file is from here. So okay, no, so I was just using this example. But you can go to any database, right? PDB is a very famous database, protein database. So I'll just simply copy and paste this. So here suppose any proteins that you guys are interested in, we can look into. Um, um, per one, that's also interesting.
Yeah, I guess I had to know possible corner.
Yeah, when I was first looking, I was like, why is this such a small search bar in the corner? Okay, so you can go from here, right? So if we go here, and then you can upload the file here, and then you can upload it from here. Okay, okay, yeah.
I have a question on the alignments and the score. I might have missed it. Like,
Yeah.
what exactly does the score like tell you? It's like conceptually wise, I don't know. It's like what is the score?
It just gives you the strength. Probably Pradeep has more experience. Yeah. Go ahead. I'm gonna let you take the question.
So Blossom kind of like a local alignment. So kind of a so Blossom 62 is something that you use when you know the two sequences. You're gonna combine and like kind of try and align with each other or very similar, like two human sequences. Then you use Blossom 62 because it takes the scoring box that you saw, like it kind of like aligns like I think I did that for you.
Okay.
It's not complicated for this before. Yeah, so let's say like multiple amino acids, like all these amino acids that we got. And so this is how the scoring pattern actually is. That is, if A is present instead of B, you get a score of 0.5. If A is present instead of C, you get 1, so on. And they have this one huge mathematical formula for creating this whole matrix. And they put that, and so let's say that is Blossom 62, and that matrix is created using that particular formula, and that is only for local alignment. And when you know that those sequences are very similar already. Uh, and PAM is for global alignment, and it's for like if you did not know, you have no idea what the sequence is, then you just go randomly use PAM. So every single time you're gonna start aligning, go with PAM, and then if it scores pretty high, go with Blossom for like segregated sequences. And the score, if the higher the score, means that the sequences are pretty close to each other, and the variance. So let's say like A, B, and C and D, and the second sequence is A, B, X, D. And when this protein is being created, like in a structure, like in a 3D structure, this doesn't really matter. Like it could be C, it could be X. The protein structure doesn't change. Functionally, it's constant. It doesn't change. Then it will give you a higher score when it's Blossom. But if it's actually giving you, like if this is protein 1, and because of X, the function changes, like the structure changes, that like some random other structure, and it's not able to function properly, then it will give you a lower score. So that is all computed with that one formula, and that's how the matrix is created, like conservative versus non-conservative changes in functions. It's a nice thing to, yeah, this is basically the how bioinformatics started.
Yeah.
Blossom and PAM.
Some pairwise to global, yeah. It's pretty, yeah. But there, yeah, this is only 4, but there's long sequences, right? So there's a lot of factors they look into, like how the protein can change.
Yeah, it should minimum have 20
Yeah. Yeah.
Thank you.
So,
Yep. I'm going to stop.